for this section, Professor Cardoso. She will talk about how the data from the MBC moved to the early breast cancer. Thank you.
Thank you very much. Thank you also to the organizers for the kind invitation to be here with you. So I'm going to focus on my talk on some overarching comments and not detail the studies again, which were brilliantly done by the previous speakers. These are my disclosures. And when I was given this title, I thought very hard what I could do. So I kind of divided the question into 4 sub-questions. So we will look at what can we learn from advanced breast cancer to apply in early breast cancer, what we thought we could learn, but then in the end didn't turn out to be exactly how expected, and what I believe that we cannot learn from advanced disease, and very briefly touch on some of the things we could learn from early disease to apply in the metastatic setting. So let's start with what we can learn from the metastatic setting to apply in the early setting. I will touch upon endpoints, making the argument that surrogate endpoints do not consistently correlate with long-term endpoints in both settings. I will also look into some of the drugs, and the take-home messages are actually there. When we see a substantial benefit in the metastatic setting, particularly if it includes an overall survival benefit, usually, not 100%, but usually the treatment becomes also useful in the early setting. But when we see just a moderate benefit, or if the benefit is only in PFS, very rarely we see a substantial benefit in the early setting, even if we may have what they call positive or statistically positive trials. And I will give some examples on 3 of the traditional standard treatments that we use, and by the end of my talk, I'll give my personal view on the potential impact of all the new drugs that have been discussed in the early setting. So let's start with endpoints. And I'll start with looking into an old paper already, looking at the surrogate endpoints in the metastatic setting, published by Martin and Pi3k when at the ORTC, looking at the early development of the RECIST criteria. And look at the conclusion over there. It says no endpoint could demonstrate, or no endpoint that was studied in here demonstrated to be a good surrogate for overall survival in these trials. Tumor response could be an acceptable surrogate for PFS. And just a little bit of history, particularly for the younger people in the audience. Initially, let's say in the 70s and early 80s, the most frequently used endpoint in advanced disease was overall response rate. But frequently what we saw is that we had a high response rate in a phase 2 trial, but then we could not confirm that in either PFS or overall survival in a phase 3 trial. And that is perhaps, or most likely, because tumor shrinkage in breast cancer is not always associated with long-term control of the disease, and very often not even with quality of life. And then a change occurred to a more time-to-event endpoints, such as time to progression and PFS, which are better as surrogates, but are still limited. So going into a little bit more detail here, you see in the 2 graphics the correlation between tumor response and survival, PFS and survival. And it shows that there is no very good correlation, which is exactly the conclusion I told you. And the authors say, except for the fact that responders were more likely to survive than non-responders. What exactly does this mean? In my perspective, it means that if there is a response in the metastatic or the early setting, that has a strong prognostic value for the individual patient. And we'll come back in the next slide to the early setting with PCR. But it doesn't mean that that has a predictive value at the trial level. It doesn't tell you that the difference in response between 2 treatments in the metastatic setting will translate in difference in survival, for example. And have we seen that in an early setting? Indeed we do, with the discussion around PCR as a surrogate endpoint. And I think there are no doubts about the strong prognostic value of PCR for the individual patient, and those who do have a PCR in their tumor actually do much, much better. However, the correlation of the magnitude of difference between two treatments and their rate of PCR does not predict for the long-term difference between those two treatments. And this more recent study is very interesting and makes the point for many tumor types. I'll guide you through it, although I don't like this mouse very much, but I'll do my best. So I highlighted in the arrows advanced breast cancer and neoadjuvant breast cancer. And look at the image just by color. If you have a red circle, it's because there is no or low level of correlation. Green means a high level of correlation, and yellow means medium. If you look at the line of advanced disease, there are really no greens, maybe 2 yellows, and most of them are red. And if you look at early breast cancer, it's the same. So actually, we don't have very good surrogate endpoints for breast cancer in terms of predicting survival. So can we use the fact that a drug is approved as a good surrogate of the benefit of that drug? I wish we could, but that's unfortunately not the case, and I'm already starting to be not politically correct in the beginning of my talk, which will make my life very difficult from now on. But if you look at this paper, we looked at 5 years of EMA-approved drugs in solid tumors, where the magnitude of clinical benefit scale from ESMO was applied both in the original 1.0 version and the 1.1 version, what you see is that with the most recent version, only 10% of the approved drugs actually provide a meaningful benefit. And this is throughout all cancers. The authors go and discuss the reasons why, and one of the reasons, or the most important reason they come up with, is because most of them were approved based on surrogates. And what do surrogates do? Well, they allow you to have a faster medicines access. They allow you to have a fast acceptance and approval of your treatment, but then it correlates poorly with the clinical benefit. Does FDA does better? No, it does not. You can look at here at a study looking at 15 years of approval, and not only the approval was not correlated with the benefit, but it was also not correlated with the cost, with the price of the treatment. Now, what about drugs? And let me start by very, very briefly and fast give you some examples. One example of chemotherapy, another of endocrine therapy, another of targeted therapy. So I went back also to another paper from Martin and Pi3k, and this was a meta-analysis of taxane versus anthracyclines in first-line metastatic setting. So what did they concluded? That single-agent taxane was significantly worse than single-agent anthracycline in PFS, but not in response rate nor OS. That taxane-based combinations were better than anthracycline-based combinations in response rate and PFS, but not in OS. So a little bit difficult to bring a conclusion, is one better than the other? And mind you that all these patients had not been previously treated with taxanes because we didn't use them in the adjuvant setting yet. Some of them were previously treated with anthracyclines. Now, what about the value of anthracyclines in early disease? This is not an extensive table. It has almost everything except the HER2-positive trials. But the point I want to make is that most of these trials were non-inferiority trials, and most of them have not proven that the non-anthracycline regimens are non-inferior to anthracycline regimens. And there are contradictory results. There's still a debate around this. We will see how we vote on Saturday. A meta-analysis is probably difficult because of heterogeneity of trials, and hopefully the Oxford overview will address this soon in one of the most the future publications. Now, I agree with the same conclusion that Martina explained yesterday and that most of us believe, that it's probably different according to the subtype and the risk. And probably in triple negative, HER2 positive, high risk tumors, let's just say it this way, the benefit of anthracyclines outweighs the potential long-term risks. But from the preclinical data and from the metastatic data, we know that these drugs are one of the most efficacious, if not the most efficacious drug, cytotoxic drug in breast cancer. So I think some more food for thought is that we have spent so many resources and so many patients were enrolled in trials, asking the question if we can remove anthracyclines, substituting for something else, but we never evaluated properly liposomal anthracyclines in the early setting, which could actually be a better solution. So how do we incorporate this with the Oxford overview data? You see that small incremental benefits have led to the benefit that we see in survival, but the trials analyzed are not trials anthracyclines versus taxanes, are trials anthracyclines versus anthracyclines plus taxanes. So I think it is very important to look at other advantages, and other advantages is that by using a sequential regimen, you use lower cumulative dose of anthracycline, and you can even lower more the risk of long-term side effects. What about endocrine therapy? What have we learned in the metastatic setting? So I went back to an old trial of letrozole versus tamoxifen as first-line therapy, and what you can see on your left, you see a small difference, about 3.4 months in time to progression. And in the right, you have the overall survival, that overall, there was no significant difference, although there was this separation of curves initially. What this translated in the early setting? The same, meaning that a very small, about 1% benefit in survival, although more or less 4% absolute benefit in lower relapses. What are the major advantages of having these two classes of drugs? Since we have to give a very long treatment, and because tolerability is also different for the individual patient, it's very good to have different family of drugs with different toxicity profiles so that we can treat the most of the patients for as long as they need. Last example on the targeted therapy. So what have we learned from Trastuzumab in the metastatic setting? As an example, the pivotal trial by Dennis Lehman, and what you can see there summarizing is that for all the endpoints evaluated, there was a substantial benefit of adding Trastuzumab, including, for the first time at that time, a substantial benefit in survival. Did that translate it in the early setting? Indeed, with a substantial decrease in mortality risk and an ESMO magnitude of clinical benefit of A consistently in almost all trials. So moving to what we thought we could learn, but it didn't turn out as expected, I'll give you two very quick examples. So we have the example of dual anti-HER2 blockade and NGS or genome-driven therapy. So let's see the dual blockade. We have mentioned this several times already, but these are the results of Cleopatra's substantial difference in overall survival. But, as said several times, most of these patients had not been previously exposed to Trastuzumab. Does that make a difference? I think we all agree that it does. When we look at the results from patients who had been previously exposed to Trastuzumab, we still see a benefit, but the benefit is smaller. However, and Martine and Rebecca Dent made this point quite a few times, saying all these trials, Marianne, Cleopatra, and so forth, are more informative for de novo metastatic patients and not really for recurrent metastatic patients. But in my opinion, that means that they should have been more informative for the early setting, which by definition is not previously exposed to trastuzumab. So I think most of us would have expected a substantial difference also in the early setting with the use of dual blockade. But as we discussed already, the result was statistically positive, but very disappointing clinically. And I'll make just 30 seconds clarification, which I discussed with Dr. Patridge. When she showed you the magnitude of clinical benefit of pertuzumab, the C was based on the neoadjuvant studies, which were very small phase 2 randomized studies. Actually, in the affinity trial, pertuzumab scores a B. Now, what about genome-driven therapy? I'll show the example of the last of these studies that was presented last year at ESMO, which is the AGATA study, just to make the point that they started, it's a very elegant study, they started with about 300 patients. They were able to evaluate 260, but then if you go down and down, in the end, only 13 patients actually received March-targeted therapy. And when you look at the benefits that they derived from that, actually only 2 patients have a durable response. So do we really need to go through all this hurdle, or can we just decide by chance and have the same result? And I'm being provocative, of course. And was this a one-time trial or result? No, as you can see here, all these efforts show the same thing. In one of the most relevant ones, the French study, same percentage, only 13% of patients actually could have a matched therapy. Is this only in breast cancer? It is not, as you can see here. This is a study looking into the benefit of genome-driven oncology for all cancers in the US, and actually about 5% of cancer patients can actually derive a benefit from this approach. I think it is disappointing, but perhaps the solution will be in technology, and the principle is not wrong, but we will see what the future holds. What I think, in my perspective, that we cannot learn from the advanced setting to apply in the early setting is regarding resistant mechanisms, or we can learn a little bit, but not that much. Because usually, not always, but usually, there are different mechanisms for primary and secondary resistance, and the advanced setting is better for a secondary resistant evaluation and the early setting for primary. One possible situation is to explore the de novo advanced disease. I'll give the very short example of ESR1 mutation, showing you that the distribution of this mutation shows clearly that the more treatment or the more pre-treated the tumor was, the biggest probability of finding these mutations. And therefore, in the early setting, we have very few of these mutations. I'm not sure it is a very useful tool there. Now, I'm going to skip this just to show you that more and more we believe that de novo and recurrent are very different situations, particularly in countries where you can give almost everything you want in the early setting, meaning that the tumors who relapse are really bad tumors who have a very bad median survival. So what can we learn from early disease to apply in the advanced setting? I don't have the time to go into those details, just saying that I believe a lot and that we don't do this way back sufficiently. Give 3 general examples. We can learn the value of multidisciplinary care, the value of experience and quality of care, to change this type of evolution, which is what we have in the metastatic setting, just the same median survival for more than a decade, to this type of evolution we see in the early setting. So a couple of examples showing you that experience, both at the hospital level and at the surgeon level, is very important. So actually, if you see here, if you have an experienced surgeon, you can have a 10% benefit in survival, which is more than most drugs can do. So the importance of having certified breast units or breast centers or breast departments, as you want to call it, and the importance of the role of, for example, Eosoma in defining quality indicators. The only point I would like to make is that those quality indicators are all related to early disease. Nobody is evaluating the quality of care in the metastatic setting. So we are now developing, together with Eosoma, under the ABC Global Alliance, indicators to certify the breast units also for the quality of care to metastatic disease. So in my conclusions, I have two sets of conclusions. One is I'm going to tell you what I think will happen to these latest drugs, and I may be completely wrong, and then I'll give you my very, very final conclusions. So what about CDK4/6? You saw that we have a substantial PFS benefit in first line, a moderate PFS benefit in second line. We still don't have statistically significant survival in second line, probably because the trials were not powered. Maybe we need a meta-analysis. So will they have a future role in early disease? My guess is probably yes, but we need to verify what's the impact in survival in first line as well. What about mTOR inhibitors? Summarizing is a moderate PFS benefit, OS not statistically significant, maybe also because trials were not powered, and toxicity was a problem, but now is being much better managed with the use of steroid mouthwash. So will they be used in the early setting? My guess is that probably not, but we will see the trials ongoing. What about Pi3k inhibitors? So far, only one show a moderate PFS benefit, all of them, in my perspective, too much toxicity for the early setting. My educated guess is that they will not make it to the early setting, but if you want to continue to study them, please focus on Pi3k mutants and not all comers. Now, what about HDAC inhibitors? I think we have now one trial showing an interesting but still moderate PFS benefit, some toxicity, and we are still waiting for a long time for this trial of entinostat. So will they have a future? I cannot tell you yet. Once we have the entinostat study, I can make an educated guess. And PARP inhibitors. So they did show a small, actually disappointing only, small PFS benefit, but a very important quality of life benefit. So I'm hoping that they do have a role in the early setting, particularly if secondary prevention proves to be more efficacious, such as we are testing in the Olympia trial. And finally, checkpoint inhibitors. From what you heard from Dr. Lloyd talk, you see that ABC and EBC are very different immune settings. And I'm not sure, or in my perspective, I think that we cannot take conclusions from the metastatic setting to apply in the early setting. We need to wait for the ongoing early setting trials. I will make a point, though, that the fact that Atezo was the approval of Atezo was based on a 2.5 months of PFS difference because they showed a result that they should not have shown, because the statistical plan said they should not have shown. I think we've been discussing if surgeons have a future in management of breast cancer. I think this is very scary for statisticians because it sends the message that don't worry about your statistical plan. As long as you can find one positive result, that's okay. You can get an approval. There you go. I've been very, very bad, and I want Richard to say if he's happy with what I just said. So final conclusions is that advanced setting is a setting of its own, has its own specificities and needs, and it should not be a quick passage to the early setting. So we have made an assessment of the needs of these patients, and we hope you can all join the effort of the Global Alliance. We can and should learn from both these settings and also from both successes and failures. And last but not least, invite you to the St. Gallen of the Metastatic in November in Lisbon. Thank you very much.
Okay, thank you. Since the time, maybe we have only one question. If there's no question, I have to close this section. Thanks all the speak. Thanks the audience. We have about 25 minutes to coffee break.